Hello and welcome to our OHBM symposium. It's my great pleasure to talk about the topic of neurovariability. Look at the brain, there's many ways we look at the brain and study its structure and its function, and that includes looking at the surface of the gyra and the sulci. We can slice through the brain and look at the sectional anatomy. We can look at connections between areas. As the connectional anatomy, and obviously we can look at functional activations and look at the functional anatomy. Now, my own journey with the brain started by looking at the surface anatomy of the brain and the connectional anatomy. So I started in the morgue looking at the brain and also learning the technique to carve out the white matter anatomy in the actual physical. At that time, a new MR imaging called diffusion weighted imaging tractography came about and actually allowed for the same idea to be applied in a living human brain to carve out or virtually dissect the white matter in the living human brain. Now, what these techniques, if we put them all together, have taught us about the brain is a general understanding or a general map of the brain of how things work and where in the brain. And I want to use my talk to discuss the neurobiology of language as an example of how important variability is. So when we look at the neurobiology of language, classical textbook description would be that there is 2 to 3 cortical areas for language functions that would be in the inferior frontal gyrus, so-called Broca's area. That's important for articulation. Then in the posterior gyrus, Wernicke's area, important for comprehension. And then there was this relatively recent addition of the inferior as Geschwin's territory. Now, obviously, these 3 regions are in 3 different, quite dispersed across the brain, but they're actually interconnected by what is known as the arcuate fasciculus, arcuate meaning arching around the lateral fissure. And you can see that in red, we have a direct connection, frontal temporal, between the inferior frontal gyrus and the posterior temporal cortex. And then we have this indirect route that relays in the inferior parietal lobe that is connecting either end in the frontal and temporal to the inferior parietal. Now, there is several different issues with this model of the neurobiology of language, and that also generalizes to other cognitive functions. And I'm going to talk you through these limitations one by one. The first one is that we have an averaging problem. Now, what I mean by that is exemplified in the slide here that was done by my intern, Eva Guzman. And what you see here is the various women who won the Nobel Prize for Physiology and Medicine. Now, if we put them all together to see what you need to look like as a woman to win the Nobel Prize, then this is a typical Nobel Prize win. Now, what you can appreciate is that even though we can probably recognize some of the features, none of the women look exactly, nor does the average look exactly like any of these women. And that is the same logic that we apply to the human brain, where we have an average brain that is very pretty and beautiful, but when we look at individual cortical areas, we can see that they do not match one-to-one onto a template, and this is exemplified here with the motor cortex, but actually applies to various different cortices. And the same holds true for the white matter language network. Now, this is obviously just anecdotal, but we looked at this on the group level. And this is the result of it. So in the study, we mapped a healthy population and demonstrated that variability is not homogeneous across the brain. And that can be seen here by the warm and the cold colors on the cortical surface on the top, and the white matter at the bottom, highlighting areas of high and low variability. Now, there's two important findings from the studies. The first is that areas of high variability tend to be the ones identified as higher cognitive function areas by neuroimaging and lesion studies. And the second finding is that for the first time, we actually show the white matter variability at the group level. And what you can see in both of these visualizations is that there seems to be a gradient whereby older the central parts of the brain tend to be more alike between us, and more lateral parts tend to be more variable. Now, this is important when it comes to applying atlases, templates, because you can assume that they're more reliably capturing the individual anatomy in the central part of the brain, but less so on the lateral aspects. What is even more important is being aware of this variability in a clinical setting. And here I just want to show you the impact that a clinical presentation of a brain tumor can actually have on the variability of the brain. And what you can see here is that the tumor displaced the cortical spinal tract. And if we were to use an atlas approach in this particular patient, then you can see in red that the atlas will predict the corticospinal tract to be in a position that it is not in this individual patient. So this is the first indication of why variability is important to be aware of. Right, coming back to the other issues that we can have with this. So we now have an idea of what the neurobiology of language looks like, but let me take you a step back. So here is what we know about the visual and the auditory system. And what you can see is it's a delicate network of boxes and arrows or lines, and each box represents a cortical area and each line represents a connection. Now, what we can appreciate here is that the visual system and the auditory system are densely packed with areas and connections, and these systems have to be extremely fine-tuned to work together. Now, if you put that in comparison to what most textbooks tell us the language in the brain, which is arguably one of the most complicated functions we can have, looks like, then it's probably fair to say that we're underrepresenting the language network in the human brain. Now, there's various reasons why we have this discrepancy. One of the obvious ones is the visual and auditory systems we can map in animals, language we can't. Now, tractography has been tremendously helpful in that because it actually enriched our understanding of what the anatomy of language looks like quite a lot beyond the arcuate fasciculus. And here's what you can see as the evolution of the language, neobiology of language from the 19th century to the 21st century. And we can see that we started with a classical arcuate fasciculus, a frontotemporal connection. Then the early 2000s, Marco Catani added the indirect segment that stops in the inferior parietal lobe. And since then, many other methods, such as direct cortical stimulation from brain tumor surgeries, have added more connections that are relevant for language in the brain. And just to show you that these are not just pretty pictures that tractography makes up, but actually we can see these connections in the postmortem brain. So there's variability in the anatomy of the brain. Now, another problem that we have with this stereotypical view of language in the brain is that we recently did a review of the literature looking at 326 studies that looked at structural variability and differences in cognitive functions in healthy controls, neurological and psychiatric patients. And there's three things that we learned from the study. The first is that most of what we know about the function of the white matter has actually been described in brain pathology. So we can see that as 45% in neurological, neurosurgical patients, and 29 psychiatric patients, only about 25% of the studies were conducted in healthy controls. The second thing that we learned is that not all brain connections are studied equally across all the cohorts. And we can see that here, for example, the corticospinal tract is very prominent in neurology and neurosurgery, but a lot less so in psychiatry or healthy controls. Now, another thing that is tremendously important about this study is that we were able to show that there is no one track, one function association. Just like it has been demonstrated, there is no one cortical area, one function association in the language relevant cortical areas. So if we look at the three segments of the arcuate fasciculus again, we can see that yes, most of the correlations were described for language, but we can see it also correlates with measures of sleep, memory, executive functions, auditory functions, attention, and motor functions. And that is not unique to the arcuate fasciculus, but in fact, we looked at all the connections and find the same pattern. There wasn't a single tract that was dedicated to one function only. So now, a great deal of variability in the white matter. The next question is, does it matter? So here's a clinical study where we worked with a stroke patient who suffered a lesion to the left hemisphere, and they lost the ability to communicate or articulate or understand language. So they became, but different types of aphasia were included. As we mapped the arcuate fasciculus in those patients in healthy right hemisphere. And what you can see here is that I highlighted three patients. The first one who did not recover language well 6 months after the symptom onset. And we can see that the arcuate fasciculus on the right side is rather small. The second patient did better and recovered slightly better with a slightly thicker connection in the right hemisphere. And then patient number 3, according to the Western Aphasia and battery, is fully back to normal. And that patient has a really thick, strong connection in the right hemisphere. Now, what's interesting about the study is that when we only look at clinical demographical data, we can explain about 30% of the variance in this population, but by adding the variability of the arcuate fasciculus, we can actually nearly double the prediction of recovery and explain about 60% of the variance in recovery. Now, another aphasia syndrome that I looked at, and the classical disconnection syndrome, so-called conduction aphasia, was first hypothesized to be due to a disconnection between the inferior frontal gyrus and the superior temporal gyrus, and that disconnection was ascribed to the arcuate fasciculus, so the red connection. That this is a classical disconnection syndrome leading to what is known as conduction aphasia, meaning that patients can articulate, can talk, patients can understand, but what they cannot do is repeat what they hear. So the information goes into the temporal lobe, but it doesn't get propagated to the frontal lobe to then repeat. Now, with the emergence of CT scanning, however, this paradigm shifted towards the inferior parietal lobe because patients with lesions in the inferior parietal lobe also presented with repetition. So what I wanted to do was to look at this discrepancy in the literature and actually see if we can disentangle it. So what we did is we took patients with primary progressive aphasia, so slowly progressing disorder, and looked at the arcuate fasciculus and the cortical region atrophy in the language networks. And what we could find was that there was absolutely no correlation between the long segment of the arcuate fasciculus in red and repetition deficits. We did, however, find a correlation between the indirect pathway and repetition, so that's green-yellow, and they relay in the inferior parietal lobe. And for the cortical areas, there was only one that survived multiple comparison correction, and that was the posterior supramarginal gyrus. Now, if we put that all together, we came to the conclusion that after 150 years, it's time to get rid of the arcuate fasciculus in the model, and we actually proposed a new anatomical model of repetition in the brain that looks more like this. Now, according to our anatomical model, a lesion can occur either in the frontoparietal or temporal part of this network and selectively affect one of the connections and still lead to repetition deficits. Now, the flavor of that deficit will be different, and that's where we see inter-individual variability between the patients and the way they present. But this model now nicely explains that discrepancy in the literature that we have seen for the last 150 years. Now, another idea that we recently put forward about the neurobiology of language is that we are moving away from a modular way of looking at the brain, whereby one area does one function. We're even moving away from a hierarchical model, which is the classical arcuate model. If we stick with the repetition example, then this would be information going into a temporal lobe and then being propagated to the frontal lobe, but really more looking at a integration in the brain where language emerges as the sum of the parts in the system. And you can think of that a bit like asking a orchestra where their music is. So if you think about it, then the music is not in the notes, it's not in the individual instrument, it's not in the individual musician, it's not in the director, in the conductor, it's not on the sheet of music. The music emerges because all of them come together in a dynamic, timely fashion. And that is what lets music emerge. And this is how we believe language works in the brain. You have all those key players, but only by integrating their function do we get something that is more than just repetition and comprehension, but really a nice complex cognitive function. With that, we have seen that variability is extremely prevalent. We have seen that the variability in the brain actually does matter when it comes to cognitive differences. But more importantly, we have seen that it does make a difference when it comes to clinical presentation and clinical recovery. So this is an indication for neurovariability being a promising area of investigation and potentially leading to new interventions and treatments and personalized approaches, patient-tailored personalized approaches to neurosciences and in particular language recovery. And with that, I thank you for your attention.